Okay. Go.
So, how do we get energy from mitochondria? In a previous lecture, I've talked about how glucose is, how the energy from glucose is harvested to get to acetyl-CoA from pyruvate. And we've made these energy carrier intermediates carrying the high energy electrons. So now we're going to do a controlled stepwise oxidation of those high energy reduced carriers. So what I hope you do today is understand the chemistry behind oxidative phosphorylation. That is, understand something about redox potential and chemiosmotic coupling. Those are important concepts. I'd like you to appreciate the stepwise flow of electrons down the electron transport chain. And I think you're really going to like ATP synthase because it's like this little motor. It's way cool. Finally, I'd like to be able to calculate the energy yield if we give you how much NADHs per glucose and so on. So we can compare fats and sugars, for example. So we've gone through food being broken down into glucose. Glycolysis takes glucose in the cytoplasm to pyruvate. Pyruvate is imported into mitochondria. It's broken down to acetyl-CoA. Acetyl-CoA feeds into the citric acid cycle, Krebs cycle, or TCA cycle. Those are all names for the same thing. Now we're going to use those NADHs to make ATP, oxidative phosphorylation. That is pumping protons, yielding ATP, oxygen combining with hydrogen to make water. So, a couple of fun words. Chemiosmotic coupling. Practice saying that at home in the privacy of your own bathroom. Sing it in the shower. Chemiosmotic coupling. So, mitochondria and chloroplasts do this. Chemiosmotic coupling. What is that? So, basically, you use an electrical potential that's created across the mitochondrial membrane, the inner membrane of mitochondria, or the thylakoid space in chloroplasts. So protons are pumped. That creates an electrical potential, a separation of charge. So you got more protons on one side of the membrane than the other, and that creates a driving force. That proton pressure, or proton motive force, is used to spin this cool thing called ATP synthase, and it's a mechanical turning motor, and that actually makes ATP from ADP. Pretty cool. Okay, so let's give you the anatomy of mitochondria first. Mitochondria are really long tubular structures, and they're snake-like in cells. When you break up cells and isolate mitochondria with the centrifuge, they look kind of like these little rugby balls. But in fact, in cells, they're in tubes, and they're constantly breaking apart and fusing with each other. They're highly dynamic. There's a lot of mitochondria in your heart and in your muscle and in your brain where you need a lot of energy, especially while you're studying cell biology. So be thankful for your mitochondria. Mitochondria have an anatomy. Mitochondria have an outer membrane and an inner membrane. The outer membrane is somewhat porous. They have these beta barrel proteins extending through the membrane called porins. And this is the membrane here. And these barrels allow small molecules through, like water or ions. Protons can get through, but enzymes are too big. So the porins allow small molecules through. The inner membrane is where all transport proteins are, and they move pyruvate, fatty acids, ATP, ADP, nucleotides, and phosphates across the membrane because they don't cross normally. They're charged. The inner membrane is highly convoluted into these cristae, and it contains 75% of the protein of mitochondria. It's where all the good stuff happens. It's highly folded, so it has a large surface area, and it has the electron transport chain, proton pumps, and ATP synthase, our cool molecule. The matrix, that's the inside. That's where it has a bunch of enzymes that are used in the TCA cycle, or the citric acid cycle. And it also has the mitochondrial genome. And it has replication and translation machinery. Why do mitochondria have DNA? Why do they have replication and translation machinery? That's because mitochondria used to be bacteria in the ancient world. And the hypothesis is that in evolution, when the first eukaryotic cells were formed, that the mitochondria were taken up into cells, and eukaryotic cells used them as part of their energy-creating machinery. So let's look at how mitochondria use the reducing power of NADH to make ATP, chemiosmotic coupling, and oxidative phosphorylation. So in words, I'm going to show you this in diagrams too. Here it is in words. Pyruvate and fatty acids are transported into mitochondria and then oxidized to acetyl-CoA. The citric acid cycle produces high-energy electron carriers, NADH and FADH2, from acetyl-CoA. And then those high-energy electron carriers are sequentially oxidized in a series of reactions. These then pump protons. So the energy is transferred from the redox reaction into an electrochemical gradient. That is an asymmetric distribution of protons across the inner mitochondrial membrane. That asymmetric distribution of charge, the electrochemical gradient or proton motive force, is then used to drive ATP synthase, which makes ATP in the matrix of mitochondria. In other words, high-energy electrons are passed from NADH and FADH2 along the electron transport chain in the inner membrane, and the energy is used to create an electrochemical gradient. That's oxidative phosphorylation, guys. We're doing redox reaction. Energy from oxidation is converted into high-energy electrons that are passed stepwise down this electron transport chain into intermediates. Each one of these steps pumps protons. And then that energy from the electrochemical gradient is used to make ATP. So the oxidative part of oxidative phosphorylation is the electrons are moving from higher to lower energy states. And each time they move down to a lower energy, we have coupling, reaction coupling, only this time it's used to pump protons. And the phosphorylation part is that the electrochemical gradient is used to make ATP. So ADP is phosphorylated, if you will, phosphorylation. So that's oxidative phosphorylation. So electron transport chain is often depicted in diagrams like this, where they show the free energy on the y-axis. And it's a series of protein machines. Many, many proteins. And I won't describe them all in detail. You're probably happy about that. The electrons are passed from the high-energy carriers, NADH and FADH2, to lower energy carriers. Quinones, I'll show you some of those. The final electron acceptor is oxygen. Oxygen is greedy for electrons. It accepts them. It wants those electrons. And you regenerate NAD plus and FAD without its hydrogen. Those are the oxidized forms. So this diagram is in your book, and it's a simplified version of what's happening. Free energy is on the y-axis. The direction of electron flow is on the x-axis. And it shows that NADH has high energy electrons. It's high on the y-axis. And oxygen has low energy electrons. It accepts those electrons. In between, there's the intermediates quinone, cytochrome C, and others. And the proton arrow here, the H plus arrow, means that during these steps, protons are pumped across the membrane, creating an electrochemical gradient. Another description of free energy is called redox potential. So redox potential is basically the negative of the delta G. So the numbers go up as they go more negative on this axis for redox potential. Redox potential is defined by chemists, and you can define redox potential for every reaction quite rigorously. So basically you have a voltimeter between two beakers with your compounds of interest, and the reference here is 1 molar protons, 1 atmosphere of hydrogen. So you measure the voltage difference between 1 molar hydrogen and 1 atmosphere of hydrogen gas and your redox reaction here, and you can get a number. Okay? It's the voltage difference between the two redox pairs. And it's a measure of electron affinity. So NADH is a high energy electron carrier. It really likes to give those electrons up. It's a poor electron acceptor, a very good electron donor. So if the redox potential is negative, the oxidized form has a poor affinity for electrons. That is, it's a good donor if it's reduced. And NADH is a good donor. That means it can donate those electrons, and they still have high energy. If the redox potential is positive, the oxidized form has a high affinity of electrons, or it's a good acceptor. So oxygen is a really good acceptor. Oxygen is hungry for those electrons. So the redox potential for the formation of water from oxygen and hydrogen is positive, highly positive. So basically you go from good electron donors to these intermediates to good electron acceptors. And the redox potential is basically, if you do the thermodynamics, the negative of the delta G. So it's the charge times the Faraday constant times the redox potential. So you can think of it as the negative of the delta G for the purpose of understanding these diagrams. So NADH on this similar diagram of redox potential going negative at the top, again it's the negative of the delta G, to oxygen goes from a good electron donor, NADH, to intermediate electron donors, finally to good electron acceptors, oxygen. Oxygen availability makes it possible to do all this stuff. So electron acceptors are good at the end, and the ones at the top, these high energy electron donors, NADH and FADH2, are good electron donors. So let's look at some of the intermediates. Quinones accept electrons first from NADH, and quinones take these double bonded oxygens, put hydrogens on those. Each molecule can take 4 high energy electrons. These quinones also flip, so when they release their protons, they transport the protons across the membrane. Iron-sulfur centers are the next electron carriers. They accept the high-energy electrons from quinones, and the cytochromes are also intermediates. So every time an electron is passed from a high-energy donor to a lower-energy donor, then the proton is pumped across the membrane. And so in this way, you build up an electrochemical gradient. And these are amazing proteins. They have some interesting structures, like they've got centers called iron-sulfur centers, and they've got covalently linked iron and sulfur in these amazing kind of inorganic crystal molecules. These are very old, highly evolved. Cytochromes contain heme groups, which are kind of like the same heme that carries oxygen in hemoglobin. These accept electrons pretty well too. And then finally, cytochrome oxidase is an amazing enzyme. It's the final electron acceptor. Final electron acceptor. It also pumps 4 protons, and it transfers the electrons to oxygen to form water. So the structure of this has been worked out. It has 2 heme groups, and the flow of the electrons goes through this copper to protein side chains to these iron and copper heme groups. So that is the oxidative part. Okay? So we've taken the high energy electrons down the electron transport chain and pumped protons across the membrane, the inner membrane of the mitochondria. So now we get to do ATP synthase, the phosphorylation part. And there's a really good movie of ATP synthase in your book's CD. It shows you that this whole lollipop head actually turns while it's creating ATP. This thing does 100 ATPs per second, or 1 ATP for every 3 protons. Protons come through here, they go through this little chain. The flow of protons turns the head. These subunits go in and out, and the mechanical energy is converted into chemical energy as a phosphate is added to ADP to form ATP. It's an amazing machine. So it uses proton motive force. That is an electrochemical gradient. Basically, more protons on the outside versus the inside of the matrix. And in fact, this thing can go backwards. If you switch the electrochemical gradient, you can cause this thing to hydrolyze ATP, and it will attempt to make the electrochemical gradient go the other way. So what is proton motive force? It's basically just the electrochemical gradient. That is the free energy for charge-ion movement. And we've talked about this before when we talked about membrane transport. The free energy for moving a charge is given by this equation, where Z is the charge, F is the Faraday constant, and V is the membrane potential. The electrical forces are large compared to the chemical forces. That is the free energy change for 1 mole of ions moving across the membrane. That is described by this equation, which derives from gas law. So for proton motive force, the Nernst equation applies. And that's very similar to the story when we're talking about how action potential works in nerve cells. So in other words, the electrical forces are large compared to the chemical forces. The concentration gradient is small, but it creates a charge gradient that is pretty much large. And so the electrical forces are very large compared to the chemical forces. So from glucose, you get 2 pyruvates, 2 NADH, and 2 ATP from glycolysis. To recap, in mitochondria, pyruvate dehydrogenase and the citric acid cycle give you 2 NADHs and then 6 more plus 2 FADH2s plus 2 GTP. So you get quite a lot of energy just from these steps. But wait, there's more. From fats, for example, from a 16-carbon fatty acid, you get 31 NADHs and 15 FADH2s plus 8 GTPs. So one molecule of fat, 16 carbon, gives you a lot more of these than does glucose. And then these will be used to go down the electron transport chain for oxidative phosphorylation to make a lot more ATP. So each NADH can produce about 2.5 or 3 ATPs, and each FADH2 can produce about 1.5 when oxygen is present to be the electron acceptor. This all requires oxygen. So that means one glucose completely oxidized to CO2 leaves about 30 ATP, yields about 30 ATP. So you can do the calculations for one 16-carbon fat molecule. How many ATPs are you going to get from that? This whole thing is a pretty efficient, it's a lot more efficient than your car. Energy is harvested with about 40% efficiency. The rest is lost as heat. But your car is probably less. A good wood stove, as I recall from wood stove purchasing days, is about 20% efficient in harvesting heat. So your cells are a lot better than that. Cells are pretty smart. Thank you.